Human cells have ribosomes, which translate human mRNA into human proteins. So human proteins are produced from the instructions in human mRNA at human ribosomes. Coronavirus hijacks this and inserts its viral mRNA into human cells and allows the human ribosomes to produce viral protein from this viral mRNA. If the human immune system is to develop immunity against coronavirus, it must learn to recognize and attack viral specific proteins. The most obvious candidate, although there are a number of proteins produced by coronavirus, is the spike protein, which in these animations I've made red because these cover the outside of coronavirus, and it is these proteins which bind to the ACE2 receptor. So human antibodies which bound to the viral spike proteins would not only identify the particle as foreign, it would also then potentially block these spike proteins from binding to the ACE2 receptors and entering human cells. How might a vaccine work? Well, a traditional vaccine might try to generate a mutant strain of coronavirus which could not cause disease. It would be non-virulent, and therefore you could learn to recognize the virus without the danger of being exposed to something which is infectious. However, there's the potential danger that the virus could mutate again, or that the vaccine could be improperly prepared, and that you could inject the host with a virus which is actually capable of causing disease. These traditional vaccines would also take much longer to develop and test. Another way of preparing a traditional vaccine would be to create antigens, such as the red spike proteins of coronavirus from the pathogen, and then inject that into the host so that the immune system would learn to recognize these proteins from coronavirus. The potential danger is that the vaccine might not be prepared properly, and so therefore the vaccine might not provide immunity. For example, if the proteins denatured and they were no longer efficient in causing antibody production against them. This would also take longer to develop since the proteins would have to be produced, purified, and maintained. In 2020, a brand new type of vaccine was developed, one that did not use a non-virulent strain of the pathogen or the proteins from the pathogen, but instead one which used RNA. RNA, if it can be injected into a host cell, could be used to make proteins from the pathogen inside the host cell, so they would not have to be produced elsewhere, stored, purified, etc. The human immune system could actually encounter human cells with the protein from the virus, which speeds up parts of adaptive immunity. This type of vaccine using RNA was demonstrated for the first time in 1994 and eliminated the need for the production of the virus antigens, since it's the host cells producing them, and eliminates the chance of infection since the host is only making a protein, not the entire virus. Also, the vaccine could be developed much more quickly and have a much shorter experience in trials, since there would be less chance of adverse reactions. The first two vaccines approved for coronavirus were RNA vaccines.